you very much for the invitation and for the interest in quantitative phase imaging. As was mentioned, I'm a faculty in electrical engineering at Urbana-Champaign, and I'm here to talk to you about SLIM and GLIM, two methods that Phi Optics is that are developed in my lab that now Phi Optics is commercializing. And I'd like to first acknowledge the contributors to this work. So on the left-hand side, you see part of my group, especially those highlighted contributed to these slides. On the right-hand side, you see a long list of collaborators, probably incomplete, without whom we couldn't really do a lot of our work, especially in biological applications. So we collaborate with people from engineering, but also from molecular cell biology, chemical engineering, animal science, even pathology, and some of our collaborators now moved to different campuses. So the outline of my talk is as follows. I'm going to start with some background and introduction and basically define what quantitative phase imaging is and the motivation for it. Then I'm going to present, as I said, SLIM and GLIM, two kind of sister techniques, but at the same time complementary in some ways. And a recent version of SLIM that works with the stained tissues, which I think opens up a lot of applications. And I chose a more recent application that is yet to be published on myelin imaging, which is, in my opinion, very exciting. Something that has been achieved for the first time in my mind, quantifying myelin at the single axon level without labels. So let me start with the background. I think hopefully you're able to see the movie as well. This qualifies or motivates most of our work in the lab. The fact that even though fluorescence microscopy has been the main working horse in cell biology, is not without limitations. And what you're looking at here is some live cells that are tagged with fluorescence, 2 channels in this case. And you see that after a while, they basically become very unhappy and these cells are dead and eventually all of them are dead. So this is part of what we call phototoxicity. So sometimes the dyes are toxic. All the time, the light that excites the fluorescence, usually in the UV, is actually toxic to the cells and it affects them and eventually kills them. There is another limitation with fluorescence that is called photobleaching, meaning that after you continuously image the cells for a long time, after a while that fluorescence quenches and disappears. So it means that you have a very narrow interval of time or window of studying these cells before actually the fluorescence goes away. So our solution to these problems is to provide a map that is entirely intrinsic in terms of contrast, meaning we do not add any tags or stains from the outside. And therefore, first of all, there is no photobleaching. Nothing is photobleaching here. You can image this forever. And it's also non-destructive. There is no phototoxicity because the white light levels are very, very low. And they could be visible or even infrared, which is a lot less toxic to the live cells. So this, in addition to having an intrinsic form of contrast, these numbers here are actually meaningful. So they mean optical path length. So how long, kind of a time delay that the light took to propagate at each point in the field of view. So these are nanometers of optical path length. So actually, you can convert this optical parameter, which is the phase delay, into something like density of the cell, and therefore connect to something that is now biologically relevant, how cells grow, how they produce mass, proteins, lipids, and so on. So non-destructive, there is no photobleaching, very low phototoxicity, and on top of that, we can quantify these biophysical properties like it wasn't possible before. So basically my talk is about the power of imaging with phase, and this is an article that I wrote a couple of years ago. For those interested, there is also a book that now it's been a while since it came out, but this was summarizing where the field was at at that time. And if you are more comfortable with this, there is a version of it from a couple of years ago. And if you like books, and since I don't know what to do with my free time, I'm working on a 10-volume series on biophotonics, much larger kind of opus. And only the first 2 volumes are out. I'm working on the number 3 and hopefully 4 this year. So keep an eye on this. And I'm not showing you these 2 cell books, by the way. If you're interested, I'll send you a PDF if you send me an email. Okay. So let's get back to a little bit more technical discussion. So if we want to extract this phase map or delay map through a piece of tissue or cells, there are 2 main geometries that can be used. First of all, both of these approaches involve interference. So in other words, you need to use more than 1 beam of light. Interference means combining at least 2 different beams. One that went through the sample and one that didn't. So in other words, they compare 2 different wavefronts, one that was disturbed by the specimen, one that is kept undisturbed, and that's why we call it a reference. Only then do you get this wavefront or phase information that otherwise gets lost. So if I don't have a reference beam here, I will only measure an intensity that has no phase information whatsoever. So there are 2 geometries. One, when the reference is parallel to the imaging field. So imagine you have a microscope, you create a light replica from the specimen to the camera, to the image plane right here. I come with a reference that is parallel to that, and I control the phase delay between the reference and this imaging field. So in other words, when you look at the interference, what you get, what the camera sees, is the result of an equation that has too many unknowns in it. So it has the intensities of these 2, the amplitudes of these 2 beams, and cosine and sine of the phase difference between the 2. So obviously you cannot extract the phase just from a single measurement. So with this phase shifting method, what you can do is to add increments of pi over 2 here. So you convert those cosines into sines and so on. So you generate basically more data such that you are able to extract the phase uniquely. So you eliminate those amplitude unknowns, roughly speaking. The second version is an off-axis method, in which we have a reference that actually is tilted at an angle. In this case, the interference is actually encoded spatially. So now my phase information is how these little fringes are actually bent. Much like you see ripples on the surface of a pond that may get disturbed by some kind of object. It's kind of the same idea. The processing takes place in the Fourier domain, but that's kind of a technicality. In the end, you get the same phase map from a single image, as opposed to this phase shifting method where we need to shift measure 3 or 4 times. So obviously this is slower, well, this is much faster. But there is no free lunch. The benefit of the phase shifting one is that gives you a higher resolution or higher field of view, while here you have to compromise one of them. You pick which one you want to compromise, but essentially you are throwing away some pixels because you need to sample this interferogram very, very well. No matter how you do the experiment, the main challenge, and in my opinion, the reason we don't have quantitative phase microscopes everywhere in every single biology lab, is really noise. And this, for some of you who are familiar with the Michelson and Morley experiment from 1887, they were trying to detect the presence of ether by measuring the speed of light on 2 perpendicular directions. Basically, this Michelson interferometer and what we are doing today is measuring the exact same thing. Phase shift. That's how they try to detect the presence of ether. And what they had to say is that in the first experiment, one of the principal difficulties encountered was the system's extreme sensitivity to vibration. This was so great that it was impossible to see the interference fringes except at brief intervals when working in the city, even at 2 o'clock in the morning. So when you combine 2 beams and they go on 2 physical paths, going through different optical components, passing through different air, different atmosphere. This simple difference actually creates random noise that washes out this phase information to basically unmeasurable levels. In other words, it takes very little to create a noise level of the order of the wavelengths of light. So for visible light, imagine that's half a micron of delay. So if you breathe on top of your experiment, if there is a vibration in the mirrors or lenses and so on, that information is gone. The exact same problem is in the much newer microsound interferometer, which probably you have heard in the news, the LIGO project, a multi-billion-dollar project that was subject to the Nobel Prize a couple of years ago for detecting gravitational waves, is the exact same interferometer except an exaggerated version. Now the arms of the two microsound interferometers are of the order of 4.7 kilometers, something like that. So the only reason it costs billions of dollars, aside from making these little subways here, is because you need this kind of stability down to 10 to minus 9 nanometer, if you can picture such a thing. So all that budget was spent to actually stabilize this, and then with passive stabilization, active stabilization, they've actually vacuumed this mini subway completely. So all this craziness went just to stabilize, to reduce that noise that was in the interferometer. Our idea, since we don't have LIGO's budget, is to actually make something that is intrinsically stable. So obviously it's much better if you can remove the noise from your detection before you actually detect it, rather than measuring a noisy signal and then remove it. So both these methods, SLIM and GLIM, are actually what we call common path interferometers, where the idea is to have the two beams travel on top of one another as close as possible. So they see the exact same noise, same vibrations, same air fluctuations, such that the difference between the two phases is very stable. So each of the interferometer arms is noisy, but the noise is now the same, as opposed to in a microsound interferometer where they are split and they have uncorrelated noise. So here's the idea with SLIM. This is a commercial phase contrast microscope. And basically, we add a module at the output of the microscope. So this is the image plane where the biologist usually adds a camera, and that's it. That's the end of their story. This is basically where we start. So if you don't know how the phase contrast microscope works, this was a Nobel Prize winning idea from 1953 that Zernike received. That was the Nobel Prize award. He invented this in the 30s. Mid-30s. So basically the idea is to have a filter in the back focal plane of the objective. So if you ever held a phase contrast objective in light, you will see a dark ring in the pupil. Essentially that ring corresponds to the incident light. So the illumination light is also shaped like a ring. It comes from the aperture of the condenser. So imagine that the incident light is all focused along this ring. And Zernike created a thin layer here that shifts that incident light by pi over 2. So essentially what he did, if you remember that equation I mentioned earlier, where you have a cosine, he converted it into a sine. So for small arguments, if you remember from high school, the sine of x is approximately linear in x. So it's good. The intensity is proportional to the phase. If you leave it as a cosine, cosine is actually 1 minus, cosine of x is 1 minus x squared. In other words, a much lazier function at the origin. So if you plot the cosine, it looks like a parabola, upside down parabola at the origin, while the sine is a line. So basically, if you convert the cosine into a sine, you get an intensity that is much more sensitive to that phase shift. So that's what this little ring does here. And that's how the Zernike got the Nobel Prize. All of a sudden, you get very high contrast in transparent samples just by converting a cosine into a sine. The problem is this map is qualitative. It means it gives you an intensity, but you cannot extract the phase information the way we want quantitatively. Because remember, you have too many unknowns. You have the two amplitudes that you don't know. You need to generate more data. So with this idea in mind, we wanted to apply the phase shifting idea that I mentioned earlier. So in other words, we created a ring that is tunable. So instead of being fixed at pi over 2, the way Zernike left it, we projected it with this lens onto a liquid crystal modulator. So this is we relayed the plane of the ring onto a liquid crystal modulator that is able to shift that in increments of pi over 2 or any other increment. So without anything on the liquid crystal, it acts as a mirror. The light bounces back, the beam splitter sends it back to the camera, and I get Zernike's original phase contrast. But the moment I add pi over 2 more, I get another image here. And then pi over 2 more, I get another image, and you see they're all different. So with these 4 frames now in test images, I am able to decouple all those unknowns uniquely. So I combine them in a very easy way, and I get a quantitative phase map that is now free of the amplitudes and all the other unknowns. So the important thing I kept telling you about the common path, if you look here, there is no second arm that goes somewhere else, like in a microscopic interferometer. What interferes here is this light that is focused here on the ring, and the light that is spread outside the ring. And these are almost perfectly overlapped, meaning the vibrations, there is only one set of optics that both of them goes through. The air is about the same. And basically that's why it's very, very stable. So we tested this initially when this paper came out, when SLIM came out. So we compared SLIM with AFM, atomic force microscopy. So we looked at the carbon film that is about 4 nanometer, has a 4 nanometer groove step. And they agree within a fraction of a nanometer. It turns out that this is actually the spatial noise in our system. So if I measure nothing, I get the Gaussian of about 0.3 nanometer. So that's the ultimate limit for my phase measurement based on my phase measurement using SLIM, which is actually pretty good. There's a lot to be discussed here. We use the white light from the phase contrast microscope. And because of that, we have no speckles. If I use the laser to do the same thing, I'll get a background that looks kind of like this. It's at least an order of magnitude worse. So white light is very good for imaging. It gives you a very, very uniform background. If I measure the temporal noise, it's even better. So in other words, if I measure over and over and over many, many times, I'll get a Gaussian noise that is it's 10 times narrower. This is the common path that I kept mentioning about, that this is actually intrinsically stable. It's not LIGO stable. We need a few more orders of magnitude here. But on the other hand, it's kind of the same orders of magnitude cheaper than the LIGO system. So it's much better actually to detect, to remove the noise before you detect it rather than afterwards. Another interesting thing and valuable for us is that this module attaches to an existing microscope that looks already like this. This is an instrument that the biologist is already familiar with, has used it for many years maybe. They have experiments built around it. Importantly, you have all these accessories that keeps your cells alive. You have an automated scanning stage or whatever you're used to. None of this goes away. Basically all the interface and accessories is the same. We just add our module here, and we convert it into something much more powerful. Also, because the paths overlaps, I can get SLIM and fluorescence on the exact same field of view, like for example here. So you look at the CHO cell, and this fluorescence is actually GFP is expressed in the mitochondria. So basically the idea is, if we want to get specific measurements, we could do this, go back and forth between SLIM and fluorescence. And once in this case, you see how the mitochondria looks, you can actually go and recognize in the SLIM image that actually these little sausages are in fact mitochondria. So you can in principle create a digital map from the fluorescence and use it back into the SLIM and get phase information and density information straight from the SLIM image. By the way, you noticed the photo bleaching, right? At the end, when the movie restarted, you see a much brighter image. That's photo bleaching. The sensitivity is incredible, I mentioned to you, especially because of the lack of speckles. So because of that, we're actually able to, we proved that we're able to image single microtubules on a piece of glass. So these are 24 nanometer diameter structures, integral to the cell cytoskeleton. And of course, they have been studied only with fluorescence, as far as I know. This is the SLIM raw data. So just imaging with no other preparation, imaging them on a piece of slide, you get this. If you do some averaging in the time domain, really the signal to noise becomes really much better. So these are 24 nanometer little tubes on a piece of glass without staining. I think they've been imaged here for the first time. Now, talking a little bit about the color bar, we showed many years ago that actually, and we're not the first to show that, but I think we're able to do much more, with much higher sensitivity over longer periods of time, that you can convert phase information into growth. And you look at the Nicoli cell, so bacterium that divides, and then you can track them over arbitrary time points, time intervals, and convert just by imaging. It's as if you're weighing the cell and you convert it into dry mass of the order of picograms, which is really very high sensitivity. If you look at the fixed cell, fixed bacterium, we found that actually the noise, which is the ultimate limit of my sensitivity, is about 20 femtograms of dry mass. So this is an incredible type of sensitivity that you wouldn't get with any scale. There is no scale that would measure such numbers. Roll ahead a few years, more recently we looked at applying this phase map to convert into density map and growth, and we looked at neurons. So you're seeing very sparse neurons that are trying to find each other, as you can see here. Then an intermediate density network that is starting to form, and then a fully formed network where the neurons already kind of migrated, made the connections, and basically just by looking at it, you can tell they don't need to grow anymore, as opposed to this sparse culture where actually they are producing a lot of protein just to connect to the neighbors. So we wanted to quantify this. Again, I don't think this has been done before, over long periods of time. And we did this over a few days. And you notice what we're trying to achieve here. So we're trying to quantify, this is a long axon, by the way, this is the cone at the end of it. And you see how much they have to produce and kind of grow and look for chemical cues that will connect them with some neighbors. And that's how the networks are made. That's how probably we remember things. Okay. So what we did, we imaged these over long periods of time. Then there was a kind of a painful procedure to track all these neurites, dendrites, and axons, and then convert this phase information into mass information. So long story short, we were able to show that actually this sparse culture grows about 5 to 6 times, maybe 7 times faster than the fully developed network. So again, the network doesn't need to grow anymore. They're already connected. They spend a lot of their energy actually exchanging information, because we can understand also the transport, the mass transport between these nuclei, between these neurons. So anyway, I think this is the first time that something like this can be done to really monitor a neural network as it grows over many days without killing it, without toxicity, without affecting its function too much. This is kind of a parenthesis, if you like. The Nobel Prize winning idea of phase contrast actually comes with an artifact that everybody knows, and it's called halo. So the idea is that because this is kind of a technical discussion, but because that ring where the incident light is focused is not infinitely thin, because it has a finite thickness, it means some scattered light, that's not the incident light, also gets focused there. So it gets focused in the wrong place, so to speak. So biologists basically lived with it for decades. They even use it when the cell divides, actually you see a more pronounced halo. And basically it is an artifact that propagates into our SLIM measurement, because we're using the original phase contrast and we add to it. So for years, we didn't care too much about this phase contrast. We kind of, about the halo, we kind of went around it, we thresholded it when we tried to quantify things. After a while, it became kind of a nuisance, and basically we fixed it. And not only did we fix it, we made it a real-time operation. So this is actually the interface of the SLIM system that is commercially available. And this is the original SLIM coming from phase contrast, from Zernike's halo. Then you push a button and it's numerically reversed. Just because we have access to that full information, we have access not to just one frame, we have access to everything that is in that image, the full 4 frames. So anyway, it was an interesting problem that combines scattering with coherence and so on. So as a byproduct, we kind of fixed Zernike's halo from 1935. Another very, very valuable thing is that just by simply scanning the cell or the specimen through focus, you are able to see the 3D structure basically like this. So we knew this from the beginning. This is looking, for example, at the neuron again in culture. You see the top of the cell, you see the bottom. So you see here out of focus. Then inside the cell, this is the nucleolus in the center of the nucleus. And little by little, everything comes into focus. But at the same time, we were clear that we have contributions from autofocus. There is blurring that goes on in the z-axis. So basically, getting holographic data to extract 3D structure of the object is an idea that was proposed early on by Emil Wolf, pioneer of optical coherence. Maybe some of you know of him. With Born, he has this book on optics that has been referred as a Bible of optics for many years. So basically, this paper is a pioneering paper that set off the whole field of diffraction tomography. So based on Gabor's holography, Emil Wolf proposed a theoretical model to use that holographic data to extract pure 3D structure object information, which means if you can do that, basically you are getting just the pure structure of the object without the effects of the measurement, the system, or without the artifacts of the imaging microscope. The problem is we couldn't apply this method because this was, number one, done for monochromatic light, and number two, for scattering light, far field measurements from the object, as opposed to what we're doing, which is high resolution, high angle, high NA imaging of the object. So we had to do these two generalizations in a way. And I have to say, it took us a long time to get it to work. We didn't know how to do it for a while. But finally, we got it to work, and basically, this is kind of the model. The reconstruction model is captured here. Essentially, this is the object we are trying to reconstruct. So imagine this would be the refractive index as a function of x, y, z. This is the data we measure. This is what SLIM gives us, and this is the blurring function. So the blurring function has a numerical aperture term, and then it has a spectrum of the light term. Basically, I'm just going to leave it at this. You need high numerical aperture and wide light to make this work. The reason SLIM works for tomography is because we have both of them. If you use high NA with the monochromatic light, it's not going to work. If you use wide light with zero numerical aperture, it's not going to work. It's a lucky kind of situation that actually works because we have both. So I'll leave it at that. If you know that reconstruction model, then you can go ahead and take away that blurriness, most of it out, and reconstruct tomograms of cells that look like this. This is a map of now refractive index. So you don't need to use dyes, you don't need to use stains, you don't need to fix the cells. This is actually a live, happy cell that we can image for a long, long time. Here's another example of a couple of neurons stuck together. So this is simply scanning through focus and applying that mathematical model to reduce the blurring. Okay, I have some good news. Last week, we have this paper that just came out in Nature Photonics. This is a step further in the diffraction tomography field, I think. We extended the diffraction tomography to second harmonic generation. So you know, second harmonic generation microscopy has been an established method for looking at, especially at anisotropic structures, such as collagen, for example. And it is a scattering method. It is a coherent method, meaning the phases are kind of meaningful and they're preserved. So it's a coherent interaction between the light and the tissue. And therefore, we had this idea, how about can we come up with a physical model that will give us a 3D object structure in terms of response to the chi2 or second order nonlinearity response, can we do it the same way as we did it for the linear interaction? So I'm going to skip over a lot of the details, but the mathematics gets a little bit more complicated, but now in the meantime, we got a little bit better at it. I wanted to point out the sectioning is really, really good. So this is kind of a frequency domain. You know, the fatter this kind of cone, the better for your sectioning, roughly speaking. So at the end of the day, you have the same idea that from just second harmonic. So this is, imagine you come with the infrared photon and you get out green photons. So you generate twice the frequency. And normally you get fields. You get the intensity of the field or the phase of the field. You don't get the object itself. So what we provided here is a way to go from the data, the measurement of the light, to the pure structure of the object, which is essentially solving an inverse problem. So you see, you go from this autofocus kind of object to a clean object. As you scan through focus, you see it's a lot cleaner and it looks kind of, the tomography looks something like this. So I think this is for the first time that anybody solved an inverse problem with holographic data for second harmonic. I just wanted to mention this very briefly. Makes me very happy that we did it 50 years after the original paper by Emil Wolf, which is like a nice way to kind of credit Emil Wolf as a pioneer. Talk about 3D imaging moving forward. A few years ago, we got these samples from Animal Science from a collaborator. He said, you know, I read about SLIM and it's great. You can do label-free imaging. I have the perfect application for you. In vitro fertilization. We want to select embryos with high viability. And obviously, I cannot use fluorescence markers and then implant it in their case, they implanted in cattle. But the same problem is for humans. You cannot add chemical markers and then select and implant in the mother because the functionality will be affected. So we were very optimistic about the outcome, but the problem is once we use SLIM to look at this embryo, you see the contrast is terrible. So the image quality is really, really not good. And we were very disappointed for a couple of days, and then we realized what the problem was. The problem is that light that gets focused onto the ring, the incident light, as you add more and more thicker and thicker objects, and this embryo is an order of magnitude thicker than a single cell, that incident light in strongly scattering media actually decays exponentially, basically it disappears. It's like, for example, if you are in an airplane and you look at the cloud, the reason you cannot see through the cloud is because the light on the other side gets scattered too strongly. It basically diminishes exponentially. It's barely there anymore. So all you see is this multiply scattered light that makes the cloud look white. The exact same thing happened in an embryo at a much shorter scale, but the idea is the same. So basically, at the end of the day, in my interferometer, I have the two beams that interfere. Remember, one was the incident light that now is very low, and the scattering light, which is now very, very strong. And the principles of interferometry say when you interfere a small field with a very high field, you get very, very low contrast of interference. Low contrast of interference means for me, low contrast in my image. So that's exactly what we saw in the previous image. So this motivated us to come up with another idea where the two interfering beams are equal in power, even though they get degraded exponentially, they go through the same cloud, but they remain equal in power and they interfere with great contrast. So basically GLIM is doing to a DIC or Nomarsky microscope exactly what SLIM does to a phase contrast one. So for those who don't know, in Nomarsky microscopy, you have two replicas of the same beam, of the same image, slightly shifted and overlapped. So essentially, when you look at the difference between these two images, looks like the difference over function minus function shifted, that's approximating a derivative of the phase. So that's why we call it gradient, because it extracts a phase derivative, not the phase itself. This derivative we can integrate later, but what comes out of the instrument is a is a gradient. So you see here, this is a negative gradient, this is a positive, black, bright. So the idea is the same. We have a module, we control the phase shift between the two. It's actually simpler because we don't need to create a ring or any other mask. It's simply a piston that shifts up and down with respect to one another. But again, it's the same thing in terms of acquiring 4 frames, combining them, and I can integrate it. This is a bit, this shows that actually this works quantitatively. Our software is also integrated and fully controlled. It's fully automated. So you see the GLIM combining the 4 frames, you get a perfect high contrast image. Actually, you can get the scattering information too. That's kind of a technicality. So now with this new instrument, we can do tomography even better than before. So this is single cells. This is because now GLIM can work with fully open numerical aperture. In other words, you add higher frequencies or you add higher Z resolution to your imaging system. But really where it shines is when going back to thicker objects like embryos again. So remember, this is how SLIM look like. Now this is how GLIM looks like. So it's as if you took this blanket of blurriness out of the structure. You start to see these little lipid droplets that are about 1 to 2 microns in diameter. So it's really almost like magic. But we understand very well what happens is that now the two interfering beams are equal in power. So by shifting cosines into sines and so on, and subtracting 2 by 2, you subtract that multiple scattering light, that cloud light that doesn't shift, that acts as an incoherent background. And really, this is what covers normally the contrast in your image. Anyway, so we're still working on the application on this. So we can image now embryos over many days. The 3 to 5 day period is actually critical for implantation in IVF. And you see, for example, here, this embryo is doing fine. These are days, hours, minutes. It's doing great up until about the 4th day. And then boom, something terrible happened here. You see basically the embryo died. And the whole challenge here is how can we predict this event before, hopefully before it gets implanted in a mother, because you don't want this. The worst thing that can happen is to do the implantation, go through the whole process that also costs $20,000, $30,000 for humans, and then have this happen inside, and this is not a successful pregnancy. So we need to do this with the intrinsic markers without adding chemicals from the outside, but I think the opportunities and the payoffs are really great here. And by the way, on the cattle industry, most of the cows I was told in the US, 90% of them come from a handful of bulls, if you can imagine that. So IVF IVF is really widely spread in the cattle industry. It's really a huge multi-billion-dollar industry. And we've published also on sperm characterization. I don't have time to talk about it, but again, the idea of label-free is really important for IVF, whether it's humans or animals. It's something you don't want to mess with the chemicals before the procedure. So I was going to show here that you can actually make these images really nice. You can render them, and then counting the cells, you can quantify their size and mass. Cell count is actually one parameter that goes into the viability. So the normal embryo has to have checks in time. So at this moment in time, you should have this many cells and so on. So this could be wrapped into our intrinsic viability marker. One exciting application that we've been working on is to look now at brain slices. So these are models for neuroscience that are very powerful. They're extracted from the animal between 300 and 500 microns, and they can be kept alive for a few hours, meaning that you have access to actually full connectivity as opposed to the neurons I showed you in a dish. This is completely different, meaning a lot more complicated, a lot more challenging to image, as you can see here. So it's a lot of background, a lot of scattering. This is again like a cloud problem. But you see with GLIM, we actually can remove that and we see individual neurons and sometimes subcellular structures. What you're looking at here, this is actually hippocampus. We're focusing on the hippocampus. This is responsible for, as you know, for the memory formation and so on. So we mounted one of these GLIM instruments onto an electrophysiology system in a neuroscience lab, and we collected data and we looked at circadian rhythms, this day and night periodicity that happens in the brain. All very interesting and completely new. Nobody's looked with quantifying cell growth, cell mass in basically a live kind of brain slice, or at least close to its natural state, at least for a couple of hours. So this is ongoing. And then we got a little bit more ambitious. How can I apply this to basically look at an in vivo piece of tissue, right? An animal or my skin, let's say, for, you know, melanoma, that's kind of a important problem. Since we have this great resolution and a way to increase contrast to remove this multiple scattering, we thought, how about we fold it and make it in a reflection instrument? So once you do that, then you are able to image bulk tissue. So I can look at my hand from this side. I don't need to go through it. So I don't need to go through this entire thickness of tissue. So actually, we managed to do that. The idea is the same. You have a modulator. We managed to actually make it smaller and less expensive, but that's kind of a detail. But the exact same idea, 4 frames, gradient, you can integrate it. But look at this. We're able now to image semiconductor wafers. So this is the ultimate kind of opaque system, right? Nothing, no visible light goes through through silicon. And in the process, we claim this is the largest quantitative phase image ever acquired, because it's something really like this big, except it's imaged with some micro resolution X and Y and nanometer scale topography. So this will take probably 2 years to do with a force microscope, and it took us maybe, I guess, less than an hour. I don't remember. So anyway, this is an ultimate example for an opaque system. If you do it in reflection, there is other benefits sometimes. For example, when you go to microplates, so in cell biology, this is very important. It allows you to do many parallel experiments, basically, and high throughput screening. Sometimes for transmission, these microplates distort the transmitted light. So imagine the ring in phase contrast gets kind of distorted. So if you do it in reflection, you have basically, you bypass all that problem. Plastic substrates are not great for DIC, therefore they are not great for GLIM either. And so if you can avoid going through the plastic, the reason is that plastic has strains in it and messes with the polarization that eventually affects our image quality. But in principle, we can do this in reflection and that problem goes away. So look at this. This is the brain slice I was telling you about, but now we're looking in reflection. So this could be much thicker now. So this slice could be kept alive much longer if it's thicker. And we go only from one side and we start to see these kind of beautiful dynamics here. And by the way, these are single cell. You can see kind of connections between the cell. And again, this is a hippocampus. So this becomes an interesting way to look at the brain slice actually over at least over a few hours. We're trying to quantify these dry mass changes, dynamics. You see, the problem is very complicated. Optically, this is really strongly scattering, but I think this is a step in the right direction. So with EpiGLIM, we're able to look at the whole animals, for example, this zebrafish, all these things that we couldn't attempt to do with SLIM. One other thing that we did more recently was to be able to image pathology slides that have been already stained. So it's kind of a funny story. I submitted a grant to NIH for using SLIM to look at pathology slides with label free, and it came back rejected with some good positive criticism, but NCI was giving grants at 7% or something at the time, and now it's up to 9 or 10. So Anyway, but one of the criticism was that asking for a slice, a pathology slice that is not stained, in other words, skipping a step in the pathology preparation, disrupts their process, slows them down. So the question was, can you do it on the already stained slices? So the answer is, of course, we can stick a stained slide and see what comes out. But the problem is how the staining actually affects our phase information. So basically, this is what we figured out in this paper here. So basically, we have a color camera now that records red, green, and blue. So it's as if, really, it's as if you're doing SLIM with 3 different colors. And it sounds kind of easy enough. It's a little bit tricky to understand the whole coherence problem and the scattering, how the 3 colors mix up, and also how to calibrate the liquid crystal. So now you have 3 colors that fall onto the liquid crystal at the same time, which is different than if you put it on a grayscale camera, believe it or not. So detecting with 3 different pixels, adding the results from the 3 different colors is slightly different than adding a grayscale camera. And the reason is that with 3 different sources, you get the coherence function modifies a little bit. You see here the difference. So what I'm trying to tell you, we looked in detail at all these effects. We call this color SLIM, and the equivalent spectrum is slightly different. But we're able to calibrate the liquid crystal. We're able to do SLIM with the stain slice. And now the first frame in the 4-frame set is actually a bright field image that looks like this. So now that reviewer was happy because we sent it back and now we got our $3 million to do this. So basically, you have all the stained tissues. By the way, I'm very happy that we solved this because I can access now all the archive tissues that are saved in the hospitals for, they are forced to keep them for at least 10 years. So we have access to a lot more easily to tissues. And the first frame in SLIM looks exactly like what the pathologist has been looking at for the past 100 years, except adding the other 3 frames from my phase shifting gives me completely new information. You see, we recover the same cell bodies. These are the nuclei, by the way. But I see a lot of stuff in here. This is called collagen. This is could be tumor microenvironment. So it's this extracellular matrix that holds the tissue together that doesn't stain very well with this HNE in here, but it shows very, with very high contrast in SLIM. So look at this here, these fibers. They don't stain very well in most of the tissue. We recover the cell body. So basically we recover everything the stain shows, except we have extra information in those fibers. That turns out that reports on how aggressive the tissue is. So we published a number of papers, both on prostate and breast tissue, that these fibers that surround the tumor are actually very informative on how fast the cancer is developing. So anyway, we're able to image stained tissues the way the reviewer asked us to do. And very happy that we did it. And I wanted to mention that this is not 2 different scans. This is the exact same scan. In one scan, I got both the HNE as the first frame and then SLIM from the other three frames. We normalize the effect of the stain. So basically, after we do this numerical normalization, there's a perfect overlap between unstained tissue and stained tissue. So in other words, we're able to get pure phase information without being affected by the quantity of the stain, which is actually a big problem in pathology. With this, I want to go to a more recent and yet unpublished study, which is to use SLIM and color SLIM. Yep.
I'm sorry, I should have been muted. Don't worry about it.
Okay. When I run out of time, please, I'm trying to be done in 5 minutes.
You're fine.
Okay. So myelin imaging is actually, first of all, myelin, as you know, is a very important piece of our central nervous system. It's a lipid-rich membrane that surrounds the axon. It really acts as an insulator to the electrical signals passing through the axons. And there are many disorders associated with lack of myelin or kind of lower myelination in the brain, associated especially with young brains in babies and so on. Internal capsule is a portion of the brain that contains a lot of myelin. This is basically a bundle, a cable of such kind of wires where a lot of information goes back and forth. So it's rich in myelin and therefore is studied in this context. We got contacted by Abbott Nutrition, the huge company that has a formula for baby food that was designed to actually promote myelination. And they have animal models that they studied with MRI that shows that indeed their baby formula works, promotes myelination. But MRI is actually not a direct measurement of myelin. First of all, resolution is of the order of millimeter at best. And you always measure actually quantity of, you measure the water content. So you have to do some kind of indirect kind of inference to get to the myelin. And fluorescence is high resolution, but you need to add labels and so on. You cannot do it live. So basically they came to us, can you see with these phase shifts, myelin being basically a fat, an oil, a refractive index that is higher than usually than the rest of the cell. Are you sensitive enough to see such a thing? So of course we didn't know. They gave us the tissues that they use for MRI, I mean, from the same kind of brains. So they had Piglet animal models. And we got 19 of these pathology slides, 900 images per slide, so a lot of data. This is how they look like. Luxofast blue is a stain that actually becomes darker where the myelin is. This is the internal capsule, so you see it's rich in myelin, therefore it's very, very blue. So the idea was to use our color SLIM to get both this and the phase on the same spot. So then from the blue channel here, I can create a mask and then put it on my SLIM and only look at that, look at the phase that comes out of that. So this is what we did here. So from the blue channel, I create a mask that looks like this, then I multiply the SLIM, and I only get basically the portion in the SLIM image that corresponds to the myelin. So we did this for the treated group, and we looked in high myelin, low myelin regions, and they had two groups of piglets, one with the appropriate for gestation. So this is normal kind of brain size, and this is small brain size. And we found huge statistical differences between the two groups. And for control diet, this is normal diet, and their experimental enriched diet for myelin promotion. So we're able to confirm what they did, but this was kind of not as exciting for us. What we really wanted to do was to look at single neurons live. And I don't know if you know this, but actually we are born with our axons not myelinated, and it takes a few weeks for the brain to connect. And the reason it does is because we have these oligodendrocytes, these specialized cells that actually come in and they start the myelinization process around the axon. So basically, what we set out to do is something I think unprecedented, to image this myelinization process as it happens at the single axon level. At the end of the day, each axon is surrounded by 20 nanometer layer of this fat. So we're looking at very, very tiny things, but encouraged by the nanoscale sensitivity, the microtubule I showed you earlier, we were really encouraged. We also needed some ground truth. So we worked with collaborators to generate these fluorescence tags. So basically we confirm that what we're looking at is myelin and not something else. Okay, so this is setting up the experiment. So you see no myelination here, and the myelinated axons, you see them in green. This is how the SLIM images look like. So we get our cues from the fluorescence, and we look here. So this is a movie that tells you what happens. So this is the axon that's getting myelinated, and these are the oligodendrocytes. And so basically they look like micro octopus that goes and wraps around and starts to shuttle from the cell body to the axon, these basically molecule by molecule to wrap around the axon and essentially electrically isolate it. So we realized that actually this network is too crowded for us, so we prefer, so we went back and designed an experiment where things are more clear. So look at these oligodendrocytes now. And this is the neuron. And this is the oligodendrocyte. It starts to wrap on this here. And then something happened. I don't know, that retracted, another one. So this is something fascinating. This takes place over a couple of weeks. So the full myelination of that 20 nanometer layer takes 2 weeks to generate. And basically, we are in the middle where, I should say, before the pandemic, we were in the middle of setting another one of these long-term experiments to confirm what we got, because what we got is this, that if I analyze single axons from my image, I am able to pick up very small increase. So we're looking at maybe a couple of nanometers a day increase in the myelin layer. And we wanted to confirm this and probably write hopefully a paper in a good journal, but we got delayed a little bit. So we are going back to the lab and hope to confirm this. But again, the idea is to measure for the first time really how the myelin builds up in a live neuron at the single axon level, which is really has not been achieved so far. So remember, I started my talk telling you about phototoxicity, photobleaching, how actually phase imaging solves all that. But basically, there's no free lunch. We gave away specificity that fluorescence is very good at. So I showed you how we built them, the instruments, so they work with fluorescence at the same time, so we can do both to get specificity back. I showed you how now we can do stained tissues for the same idea that we can get specificity. I showed you how we applied it for cancer, but also for myelin. But there is another way that is very exciting that I didn't have time to talk too much, especially since this is in the context of Phi Optics. There's a way to get specificity through computation. So I'm just going to give you a glimpse of what we're talking about. Because we can do SLIM and GLIM and fluorescence on the same field of view, we can get fluorescence, use it as ground truth to train a neural network, train a computer to do machine learning and learn how to predict fluorescence from the morphology only. And then we did that successfully. And what you're looking at here on the right-hand side is actually a synthetic fluorescence predicted by the computer on cells that have never seen dyes. In blue, you see the nuclei of the cells, and you see how it actually looks pretty good. And we have quantitative measures of how good it is. And in red, you see the membrane of the cell. So this is all through computation, through artificial intelligence. And this particular paper is under review in Nature Communications. In the meantime, we use the same idea to actually analyze subcompartments in sperm cells and actually select them for highest fertility. And this was accepted in PNS, I guess, last week or 2 weeks ago. So this is going to come out soon. On cancer, we started to use this. We have a paper out on a simple, just cancer versus normal, just using artificial intelligence that works with 98, 99% accuracy. So basically, this is a new direction for phase imaging, I think, because you really have an ideal combination now, non-destructiveness, no interference with the function of the cells, but now you get the specificity through the computation, provided that you can feed this data, ground truth, to the computer. And we have that ability because we build the instruments such that they share the path with fluorescence perfectly. So in a way, there is no manual annotation, actually. The training can be done automatically. So with this, I'd like to summarize. I told you a little bit about SLIM and GLIM and the color SLIM, and then a couple of applications. And I think the future looks really, really bright, combining QPI with computation. I need to acknowledge, of course, that I have a financial interest in Phi Optics, and this is how the SLIM instrument looks like. The GLIM can be made more compact, a version of it at least. And with this, I'd like to thank you for your attention and look forward to your questions.
Thanks very much. Thanks wonderfully very much, Dr. Popescu. Do we have questions that came in through chat? Because Catalin and Gabriel are going to do the question and answer right now based on questions which can be put in through chat.
Hello, everybody. I'm Catalin Kiritescu from Phi Optics, and thank you, Professor Popescu, for this wonderful presentation. We don't have, well, we have only one question in the chat so far, but if you have some direct questions that you would like to ask, you know, unmute yourselves. If not, we can go to Daniel Sanborn, who has a question that he would like to ask in the chat. But
okay, well, I just want to say that it's a goal of ours to have more microscopy, modern microscopy, that depends a lot on computational work. And even though you mentioned your future for more computational microscopy and the AI, it seems to me that because you showed so many complicated formulas, as far as I'm concerned, not being a mathematician or a physicist, that the technology that we're looking at is highly computational. You may not think so because you're more sophisticated. So we're really happy to have a look at some of this. I also was interested to see you imaging a microchip surface. And myself, I'm interested in art materials, and lots of times we're looking at things that are not alive, but we want to see very highly magnified the details in order to speculate about processes that are causing deterioration or structures that help art to hold its form and so forth. So for me, often this will be metallurgy, but people are looking also at different kinds of sections of stone and also parts of paintings. The traditional thing is to have cross-sectional samples that are cut for cross-sectional, but we're also very non-destructive oriented in art materials research now. Do you have any just off the top of the head comments to what I'm talking about?
I know what you're talking about. We don't have ourselves a lot of experience with artwork. I think that would be something kind of cool. I know I've seen talks and papers on various other techniques, including spectroscopy, that can be used for restorations and things like that. It's very important. I do believe, if you ask me, of course I'm biased, but I do believe this type of imaging could add to the menu of tools that are out there, especially in quantifying things. So I would imagine, again, I'm completely ignorant, but I would imagine if you want to restore things to their, I don't know, original state, probably to be able to tell how much of each compound is there probably might be helpful. In terms of metallurgical applications, let's say, if that's a word, I think the method that works now in reflection could go out of the box. Of course, you wouldn't have made it with a transmission method, but in reflection, I think we can look at all kinds of applications. I can imagine quantifying roughnesses, things like that. Topography can be done to very, very high accuracy. Rocks, again, never looked at them. The closest was looking at some roots, right? From Germany, I think we got some samples close to rocks, but still plants. I think, yeah, it will be fascinating to get some samples like that. I think for rocks, things like that, minerals, I think you can go both transmission and sometimes reflection as well, I can imagine, for some of them. But yeah, we haven't done a lot of those ourselves.
Thanks. Yeah, lots of art, especially traditional art, is made of plant materials. And as technologies have improved through time, I know I'm not young, so I've seen a lot of change here. Our conservation scientists, as they have instruments or access to instruments and training, have tried to look very closely at very small aspects of some of the materials art is made of, and it helps in their modeling of the processes, not only of making the things into art, but also of the deterioration that can happen. So it's very exciting to see what you're presenting. Thank you.
Yeah, thank you.
Daniel, would you like to ask your question?
Yes, and so my question pertains to rheology, which I assume that the members of this group would have some familiarity with, but generally it's my understanding that rheology tends to be handled by like material scientists or physicists. So if it's all right, I'd like to give people just the briefest explanation of what rheology is, so perhaps it helps to stimulate some ideas in them. So I used to work with rheological equipment, and just as a sort of demonstration, before about 15 minutes ago, this is a piece of what is essentially silly putty, and I rolled it into a perfect sphere and then left it sitting on my countertop. And as you can see, gravity, the force from gravity has caused it to essentially deform back into a flat shape.
Can I briefly interrupt you for just 1 sec? I apologize. If everybody would like to see what Daniel is showing, just simply double click on his image in the set of pictures and you'll see it magnified so you can see the, yeah, go ahead, sorry.
Yeah, so this was a perfect sphere when I rolled it into a ball about 10 minutes ago. So rheologists are interested in studying how that deformation occurs, typically with time or with force. And the way that traditional experiments are done, this almost this amount of material, perhaps slightly less, would be placed between two plates, like about the size of quarters or perhaps as big as $half. And those plates rotate to conduct experiments on how the material deforms with different amounts of force or speed applied. And of course, this is important in all kinds of material science. This is one of the basic tests that's done is to determine things like viscosity, or it might be done to determine how, for example, John, how a coating might spread out when it's painted or applied to a surface. So one of the applications that I see these microscopy techniques being able to do very effectively, and this is my question for you, Dr. Is it Popescu? Did I pronounce that correctly?
Yeah.
Okay. So of course, because I understood from Catalin that you can do very precise measurements of the morphology of this, so including all the dimensions, and because you have the refractive index data, you can typically, if you know what this material is, you can estimate the mass of this. Let's say that this was a sphere that had dimensions of a micron or nanometers. And so because you can also measure in time, then I would assume that it's possible if you were measuring this nanometer-sized droplet of precise dimensions and you know its mass and you know the speed it's traveling because you're keeping track of time, then as it deforms against the surface, I would assume that you could back out various rheological measurements from that. And that would actually be a pretty novel application because currently, as I mentioned, a lot of rheological experiments, they depend on bulk measurements. You're getting the ensemble behavior of a whole bunch of these droplets when in some cases, really what researchers want, they want to know how things behave at that a very small, that nanometer scale level or that micron scale level. And I see these techniques being almost uniquely able to provide that kind of information. Have you done any research in that area recently? I know you have in the past.
Actually, I have. So, in fact, my PhD thesis was on light scattering, static, but also dynamic light scattering, and then dynamic light scattering methods for microrheology, which is doing rheology at a very small scale. So what I showed you today, these quantitative phase imaging methods are, as you said, pretty ideal for doing microrheology, rheology at very small scales. And this is very important for biological structures for living cells, as it has been shown, for example, in, just to mention cancer, the elasticity and viscosity of the tissue is very different in a tumor versus, and in the microenvironment versus normal tissue. So the function of the cell depends on its elasticity. Actually, it has been shown that if you grow stem cells on a stiff substrate, they differentiate into bone cells, into a stiff cell, as opposed to growing them onto a soft substrate, and they become neuron cells. It's incredible, because biologists for decades thought, for centuries, that chemistry basically kind of controls everything. Turns out what you're mentioning is actually is another dimension that is much, much less explored. So more specifically, to microreology, we used phase imaging a while ago now, starting when I was a postdoc at, I started in 2002, but we have a series of papers on microreology of red blood cell membranes. So these membranes are special in the sense that they have not just the lipid bilayer, which all the cells have, which is completely viscous. It cannot, the oil cannot store energy, right? But underneath is actually, is backed by a protein mesh. That's how our red blood cells can actually deform in microvasculature to channels that are half their diameter. So they're 8 micron in diameter, and microvasculature could be 4 microns. So they squeeze and bounce back, squeeze and bounce back. So basically, our body has to make sure these cells are perfectly flexible to perform their task. So in fact, this is what the spleen does. The spleen does a microreology test to the red blood cells. They are forced through the spleen. If they cannot make it through these little holes, they're called spleen sinuses, they get stuck, and then the macrophages come and eat them. So that's how we only have 3 months old at the oldest, are 3 months in our circulation. It's really a microreology test. Now, we looked at, first of all, we developed this method to back out the rheology based from the fluctuations of the membrane. So normally, from what you measure for macroscopic objects, you apply the strain yourself with the cone and plate or whatever microscopic methods. At the micro scale, you can rely on actually the Brownian motion doing the activation for you. So because the membranes are so soft, at room temperature, they fluctuate, but the standard deviation of this fluctuation is about 30 nanometers, 50 nanometers, very small. So this phase imaging method was perfect for this application. So from the fluctuations with a physical model, we were able to get out shear modulus, bending modulus of the membranes. And then we showed that as the malaria infects the red blood cells, these moduli are completely different. So the cell becomes really, really stiff, and it basically loses elasticity, and that's how actually the cell breaks and the parasites multiply, and it's all this mechanically triggered process that is incredible. So anyway, I can talk for many more hours about this, but I think you hit on a great application for this method. We did some work. I think there is a lot more to be done.
I think this could be of interest to everyone from like polymer researchers. It could also be interesting to people who manufacture any kind of product that's eventually going to be sprayed or applied. I know that researchers who work with agrochemicals are very interested in how something like an emulsion is going to behave. For example, when it's sprayed from an airplane onto the surface of a plant leaf, and they want to know with a very high level of precision, or they want to be able to see exactly what the droplets are doing when they are striking a surface at certain
We published a
speeds.
paper on profiling droplets as they evaporate on a surface, of the wetting angle, incredible detail. None of the models that we could find applied to what we could measure, because everything was large scales. But we can actually go to 0 radius. So we can go to really small droplet scales, where we had to come up with the different theory because nothing fit there. The measurements current measurements only allow for really big, like millimeter as the smallest. One last thing I wanted to mention is also, we are able to analyze transport. So, you know, one of the methods to do microreology is to put some beads or some kind of small objects and measure their trajectories from their mean square displacement, you can actually infer the viscosity and elasticity response of the medium. But in the cell, sometimes there is no discrete point that you can track. They're mostly looking like a cloud or looking like a cotton candy, if you like, as a continuum medium. So we came up with a method that I didn't have time to show here, that imagine if the phase image is like a density map, continuous map, we are able to now extract transport information of that continuous mass. So it's not just discrete particles, but imagine it's like a fluid with inhomogeneous density that actually is transported back and forth. So we are able to decouple between diffusion, transport, and active transport, because as you can imagine in the cell, you have both. You have diffusion because you are at finite temperature, but because the cell is alive, you actually have active transport. You have these molecular motors that drag vesicles and particles from one side to another. So we're able to quantify that very, very accurately with this approach. And in principle, you can use the same thing to extract rheology.